All right, I know it's not morning anymore, but I actually still am half asleep. I will switch to a day schedule at some point. All right, I think this is where we stopped last time. We talked about lipids and lipids making up the plasma membrane, and I said that one of the characteristics of lipids, and those lipids that make the plasma membrane, is that they have a hydrophobic region. And we define hydrophobic, and even though it says water-fearing, phobic is a fear of aversion to hydro for water, it's not really water-fearing. It doesn't repel water, it doesn't push water away. It's simply that water doesn't interact very well with it. It doesn't mix very well with it. What makes the phospholipids really interesting as a molecule is that in addition to that hydrophobic region, or if you want that greasy type region that doesn't work well with water, it has a hydrophilic region. This is often just referred to as like a head group. And it's made up of a phosphate as well as other molecules of phosphate. So sometimes you'll see it called a phosphate head group. The key to that phosphate head group is that it's hydrophilic, or if you want, it's water-loving. And that means that it interacts very well with water. So this is a pretty unique molecule in that it's got an area that likes water and interacts very well with water, and it's got a region that really doesn't want to have anything to do with water. These phospholipids will self-assemble to form biological membranes, and you can actually order these things online, put them in water, put them in a mixer that sort of mixes back and forth over time, and come back in a couple of days, and you will form little spheres which superficially look like cell membranes. They self-orient so that there is water on the water-philic ends, and the hydrophobic ends, the greasy ends, interact with other fatty acid tails of other phospholipids, and then the hydrophilic ends on the other side of this are going to interact well with water. And we basically have a situation like this. This is one section of a membrane that would be part of a cell, for example. Oops. Yes, it's right here, that chunk of the membrane. Before it gets down, phospholipid tails, and the fatty acid tail point towards the middle. And you can make these things. So what's, I guess, unique about this is that this is a very structurally sound way to organize the molecules. One end deals with water really well, a whole region in the middle that really doesn't have anything to do with water, and then again, water on the inside. What are these areas of water? Well, one is the extracellular fluid, or the ECF, and on the inside, it's the intracellular fluid. What you want here, there's the intracellular fluid, and there's the extracellular fluid. And that's very stable. So what did I say I want you to think about when you think about these lipid layers and the plasma membrane, at least the lipid component of the plasma membrane? It acts as what? The barrier. And that's what really I want you to think. This is the barrier. This is preventing most molecules from entering or leaving. Your book's going to tell you something. I think, let me show you the words that the book does. It says, the lipid, and I'm going to use the word bilayer, and that's just referring to our two layers of phospholipids, is the barrier to polar or charged molecules. And it's use the phrase polar or charged molecules. Which means that molecules that are polar, do you guys remember polar from chemistry? What is a polar molecule? What characteristic does it have that makes it polar? What does that mean if something is polar? Yeah, it has a partial charge. This is really cheap. I always think of this, and hopefully you'll never forget what polar means. Imagine the North Pole, and imagine an animal that lives at the North Pole. What animal lives in the North Pole? Polar bears. Imagine the South Pole. What animal lives in the South Pole? Penguins. All right. Are there any penguins at the North Pole? No. Are there any polar bears at the South Pole? No. They're different. So the two sides are different. Top's got polar bears, bottom's got penguins. Polar molecules are kind of the same way. A polar molecule will have differences on either side, and the difference is in charge. It'll be like kind of positive at one end and like kind of negative at the other end. polar pairs and penguins. That's what it means. Or charged molecules. Well, what kind of molecules are polar molecules? Things like glucose, things like amino acids. Water's a weird one. I'm going to put this in parentheses. Your book explains that water actually can make it through the lipid component of the membrane, and it does not very well. And actually for this class, and the way that most physiological systems work, it's best not to worry about water. No, actually, it is okay. I'm just going to put an X through it, and we'll explain why. But we'll just consider water as like a separate entity and then deal with it separately. All right, what are charged molecules? What do you mean by charged molecules? Give me some examples of charged molecules. Things like sodium, potassium, calcium. Excellent. If you want, you can put chloride there. But yeah, that's a really great group of molecules. Organic molecules like glucose and amino acids, as well as ions. It's not a barrier. And your book's going to use this phrase, small, it says non-polar molecules. Instead, I make a short list. So to me, small non-polar molecules really doesn't mean a whole lot. But I can come up with two small non-polar molecules that actually do mean something to you guys. What's O2? Oxygen. And how about CO2? Carbon dioxide. Those happen to be small non-polar molecules. The plasma membrane is no barrier whatsoever to oxygen and CO2. It means that as long as there's oxygen around, it will fly right through the membrane and get into the interior of the cell. And we know that there are organelles inside the cell, like mitochondria, that need to use oxygen so that they can make ATP. Cells produce waste. They burn out things like glucose or amino acids and produce CO2. So when CO2 is produced inside the cell, it's not trapped inside the cell. It doesn't have any issues getting out because the membrane's not a barrier to it. It just flies right out. So that's really convenient and intimately related. We breathe in oxygen, we breathe out CO2. When it comes down to the cellular level, we don't have to worry about trying to transport this thing across a barrier. It just flies right through, flies right out. So there's no problem there. And then the other type of molecule is just going to be a category. Lipids. Lipids are soluble in other lipids. Oils dissolve other oils. So lipid molecules can pass right through the lipid bilayer without much difficulty. I'm just going to give you one example of a lipid that can go through the membrane easily, and we'll see it several times. Steroids. So here I've got a diagram of some steroid molecules. First there's cortisol. Have you ever heard of cortisol before? Or hydrocortisone cream is another one that you may have. You can buy that and you can rub on like things that are itchy or painful to make them stop hurting or stop itching. And then testosterone, and then estradiol, which is the most common form of estrogen. And for this class, I'm actually just going to say estrogen. I'm not going to worry about the subcategories of estrogens. I'm just going to say estrogen. Those are all steroids. Just look at the structure of it. What do they all have in common? Like 4 funky rings all interconnected. Just memorize that shape. Whenever you see something that has that shape, it's either going to be a steroid or it's going to be cholesterol. Steroids are made from cholesterol. Those are all lipid soluble. All the carbon, all the hydrogen on those molecules. And even though there's some partial positive and negative charges here and there, for the most part, they are mostly nonpolar. Dissolvable in lipids, they are lipids. So they go right through. And this actually tells us something about where testosterone has effects inside a cell. If testosterone is lipid soluble and can pass right through the membrane, when that testosterone in a person's blood gets to a cell, it doesn't have any problem going right into the cell, and ultimately it's going to end up in the nucleus, which also has a membrane, and it changes gene expression. It's one of the reasons why some hormones like testosterone and estrogen can have such powerful effects, is actually they affect the way that different genes are expressed. No problem getting in there. It goes right through the membranes and right to where it can have actions. All right. So the general rule for what goes through a membrane, I'll give it to you. Think about what does go through. Oxygen, CO2, and steroids. That goes through the membrane easily. Everything else doesn't. And that's the basic rule. And there's going to be weird exceptions here and there, but this makes it simple. You've got 3 things to remember that go right through the membrane without a problem. Everything else is prevented from going through the lipid component. Everything else is going to be excluded, at least from going through the membrane portion of the membrane. What do you think? Good? There's our barrier. Limit of the cell, lipid component. Basically, it's acting to prevent most things from entering or exiting the cell. What we call the barrier. All right, so we know that cells need things like glucose and amino acids, and that those things then have to have a way to get inside cells. We can't exclude glucose from the inside of the cell. The cell would starve. So there has to be some other mechanism or something else that the cell uses to get these molecules that are either polar or charged molecules, like ions, into or out of the cell. And those are the protein components of the cell membranes. So when we talk about cell membranes, they're made up about 50-50 by mass of lipid and about 50% by mass of proteins. So about half by mass is the barrier component, about half is the protein. Different categories of proteins in the cell membrane. Peripheral proteins. A peripheral protein is simply a protein that doesn't go all the way through the membrane. It's just stuck on one side of it. So for example, that's a peripheral protein. It's just connected to the ECF, like a little protein sticking up here. I'll talk a little bit about more of one type of peripheral protein. Mostly in this class, we're really focusing on the so-called integral proteins or transmembrane proteins. What does trans mean? Like a transatlantic flight would be a flight that goes across the Atlantic. It means across. Transmembrane proteins are proteins that extend all the way across the membrane. Good. And a protein that goes all the way across the membrane is in a position to act as a transporter. It can take things from one side of the membrane and allow them to pass through the membrane onto the other side, or things from this side and bring them across to the opposite side. Two types of transmembrane proteins, transporters and channels, we'll talk most about in this class. They're almost the same thing. A transporter is a transmembrane protein that undergoes shape changes to allow transport to happen. So typically we draw on something like this, and there'll be a little position on it where a molecule can bind, and once the molecule binds, it changes its shape. This is hard for me to draw. So sort of maybe flips its orientation, and now the molecule is free to diffuse away on the other side. So they basically act like little revolving doors. They open on one side, and then flip, and then flip, and then flip, and then flip. And that allows molecules to move across the membrane when moving through the protein. We'll refer to those as transporters. And then there are channels. Channels are simpler. Channels are more like pores. So I like to draw them kind of like a barrel. There's like a hole that runs through the middle. And this allows molecules to diffuse directly through. There's no real shape change per se. It's just a little pore, channel, hole through a protein so that molecules can go in or out of the cell through the membrane channel. Third type of integral protein or transmembrane protein is a receptor. And if we think about the types of chemical signals that a cell has to respond to, like whether it's a hormone or a neurotransmitter or something like that, most hormones and most neurotransmitters are not lipids. They can't go straight through the plasma membrane and have effects inside the cell. They'll be little polypeptides, which are amino acids. They can't cross the membrane. So in order for them to communicate with the cell, they actually have to bind onto something that's on the cell surface. And that's going to be a protein called a receptor. This will be like a little tiny hormone, something like, I don't know, insulin, and then it binds to the surface, and then something has to happen inside the cell, releasing some molecule that can have multiple effects inside the cell. But it's a way to basically think about, well, every single molecule that is supposed to be telling cells what to do. If it's not a lipid, it can't get inside the cell to carry out its instructions. It's got to bind to something on the surface. That thing is going to be called a receptor. And we'll talk a lot about different types of receptors in this class. We'll classify a few of them. We'll talk about the specific molecules that bind to it. Your book has a lot of information about the receptors. I'm going to simplify it as best as I can. So if you're reading ahead and you see what's going on, I don't want you to freak out. We're just going to break it down to the bare minimums. But this is one of the most complicated areas of physiology because there are so many different types of signaling molecules. And each signaling molecule has its own type of receptor. And some signaling molecules can bind to different types of receptors. And I don't know, in the span of like one page of text, you can go from I understand it to I'm totally lost and I will never understand it. So if you're reading ahead and you see these things, I don't want you to freak out. We'll break it down piece by piece, and I hope I can take you with me as we talk about the different types of receptors, and we'll do it a chunk at a time. But it really can be overwhelming. I think it's the, yes, I will say it is the most difficult part of physiology is all the pharmacology, all the chemicals and receptors that are involved. All right. One thing I can say now about especially these transporters and channels is when you think about something like oxygen or CO2 or those lipids, since they go right through the membrane, the cell has no ability to control whether they go inside it or out of it. The cell can't control whether oxygen is going to enter or leave because oxygen just goes right through the membrane. But with something like a transporter or something like a channel, the cell now has the ability to regulate whether or not a molecule is going to go in or out of the cell. So we might take an example of something like glucose, which is a charged molecule, partially charged polar molecule, and it can't go through the lipid bilayer, which means that the cell has to make a protein, and we'll make a little transporter that can change shape to allow it to go in. But the cell now has the ability to control how much glucose goes in. So for example, many of these transporters have little on and off switches, if you will. They can be inserted into the membrane or taken out of the membrane. Sometimes they can be modified a little bit, turned on or off. If you turn it on, the cell can use glucose. If you turn it off, the cell can't use glucose. So the cell is regulating whether it's using glucose or not. It can also regulate how much gets in. So for example, that's one transporter, but let's say the cell puts 4 in there. Now it can take up 4 times as much glucose in that same amount of time. It can regulate how much glucose is used. More transporters, more use, less transporters, less use. This is extremely powerful. All these are proteins that are coded for by the genes of the cell, which means that individual cells then have the ability to control their actions. More transporters of one type, then they can use glucose. Less transporters, then they're not going to use glucose. So this is really powerful, and the same with channels. More channels of one type, you can allow certain ions in at a certain rate, or you can use less of those and then allow less ions in. And it's up to individual cells what type of actions they're going to do. That way you can make things like liver cells, muscle cells, neurons, heart cells, and have them behave differently based on the transporters that are on the surface. Make sense? Good? All right. So the rest of this section of the chapter, we have a little bit to talk about as far as other ways that cells have to move things across the membrane. And then it's a review of the organelles. And I know you covered the organelles in a minute. I don't go through a lot of detail for organelles. I basically simplify it as much as I possibly can. Ooh, I can show you these pictures. I just stole these off the internet, but I thought they were cool. Those are channels. And these sort of like ribbons are individual amino acids of the proteins, but they show like sort of basically like a barrel-like structure. So if you would look at the protein channel, it actually makes, they almost look like those little finger things, like you stick your finger and get stuck. But those things are actually inserted into the membrane. So here would be the extracellular fluid. Here's the head groups, the phosphate head groups. There's the fatty acid tails. There's another layer of phosphate head groups, and this would be the intracellular fluid inside the cell, and it allows specific molecules to pass through those channels. Really funky looking proteins. Other ways that cells have to move things across the membrane. We all know phagocytosis. What does that mean? Yeah, cell eating. Cellular eating. And we have specialized cells that are good at phagocytosis. I think most of us have seen like pictures of amoeba in like swamps, like eating like a little paramecium and like other things. But we have cells that do exactly this. Macrophages and neutrophils. White blood cells. Nice thing about macrophages and neutrophils is, one, in part they come pre-programmed to be able to identify certain types of bacteria, and then they can eat those things. And then you may or may not have covered it in your anatomy class, but the macrophages, which used to be monocytes until they left the blood, will also ingest things that have antibodies bound to them. So they're basically specialized for looking for things that have an antibody bound to it, and if it's an antibody bound to it, it says, ooh, this must be foreign, and then can engulf it, and then eat it. Once it's engulfed, it basically has within it a little tiny vacuole, a little bit of membrane whatever it ate in. I'm just going to put a bacteria in there. What's it going to do with that thing? Do you guys remember lysosomes? As organelles? Yeah, lysosomes are little bags of enzymes. And if you fuse a lysosome with that little vacuole that has the bacteria, then all the digestive enzymes plus acid get dumped onto the bacteria and then hopefully kill it. Specialized way that cells have for getting things inside them. Endocytosis. A little less specialized. Endocytosis basically is a procedure where you make a little vacuole. We're going to call it a vesicle. So essentially, we have the plasma membrane of the cell. It can invaginate a little bit, and that invagination can get a little bit deeper, and then a little bit deeper, and then eventually pinch off, and then collect a little bit of whatever's in that extracellular fluid and put it in a little vesicle. Different ways that this can happen. Sometimes cells just do this. So there's a cell, and it just makes a little vagination, and then grabs a little bit of the extracellular fluid, and then pulls it inside itself. It doesn't actually look for anything specific in that fluid, it just takes some in. So it's going to be water, and just whatever happened to be in the extracellular fluid is now inside the cell, and it can pull nutrients out of that. Sometimes it'll actually be trying to transport it to the other side of the cell, and then it will dump it out. Other times it's very specific, so there'll be receptors on the surface of the plasma membrane that bind specific molecules. And when enough of these molecules get bound, then it starts this process of invagination, and then the invagination becomes deeper, and then you end up with whatever the cell specifically wanted to take out in a little vesicle inside. So the first type, penocytosis, it's non-specific. And the second type is referred to as receptor-mediated, and it is specific. And you can think of this basically as one way is the cell's just taking in whatever happens to be out there. This way is something very specific. There'll be protein receptors that are binding something the cell's looking for. What kinds of things could it be looking for? One is lipids. So we don't transport lipids in our blood just loose, like cholesterol, and we all heard about cholesterol in our blood. Cholesterol gets bound to proteins as sort of like a complex. It could be binding those cholesterol protein complexes, taken inside the cell, and now the cell has that cholesterol that it needed to make something like, I don't know, steroid hormones. One consequence of this that's sort of negative is that viruses, and the flu virus is one that does this, will actually put on the surface, and this is going to be way bigger because flu viruses are tiny. I'm just going to draw it gigantic. Little proteins of their own that the cell thinks is, for example, something that it would like. So it actually matches a receptor that should be bringing in like a cholesterol and protein complex, but actually that thing binds perfectly to it, and then the cell ends up taking in the flu virus, and now the cell has basically and stupidly infected itself with the flu virus. So it's specific, mostly it's good. Some viruses take advantage of that to gain access to cells and then infect them. Endocytosis, and if you break this word down, endo means into the cell. So it just means taking things inside the cell. Exocytosis. Exo out of, or in other words, we're taking things and we're exiting them from the cell. Exocytosis relies on vesicles. So the cell has to produce little vesicles, they're membrane-bound structures. And inside the vesicle is going to be some kind of product. This will often be a protein product, something that the cell wants to secrete. Examples of things that can be secreted, things like hormones. And not lipid hormones, but things like protein hormones, insulin, for example, is a protein-based hormone, specific hormones. Things like neurotransmitters. So when a neuron releases chemicals from it, it actually stores those chemicals, those neurotransmitters, in little vesicles. And even other sorts of secretions, like I'll put sweat up here. So your sweat glands, when you make sweat, you actually store the sweat in little tiny vesicles. And then those vesicles get lined up along the plasma membrane, and they're basically waiting for a signal. It could be an electrical signal, it could be a chemical signal, but upon the signal, they will connect with the plasma membrane, and then whatever's inside the vesicle gets dumped out. And your book has a more fancy description, talking about snare proteins and things like that. I don't care about that. You can just say that they connect to the membrane, and then whatever's inside that vesicle now got dumped out into the extracellular fluids. This is the main way that cells secrete things. So when we talk about endocrine glands, this is mostly what's happening. We have endocrine cells secreting something, some hormone. This is how it gets out. This is how all neurotransmitters release, or all neurons release their neurotransmitters. They got to produce these vesicles, line them up, and then have extracellular happen, fuse the vesicle with the membrane, and then dump those things outside the cell. Cilia and flagella, I just want you to read, where did I put it? Just read over these things. I expect that you remember what they are. But we'll talk about them in lab, and you don't need to worry about them now. Cytoplasm and its organelles. This is our review of all the organelles in the cell. And I'm not going to do more detail than you did in your anatomy class. We need definitions. What's the cytoplasm?
Where the organelles are held,
What's that?
where the organelles are placed.
How about this? Yeah, inside of, there we go. Everything inside the cell, except the nucleus. So this definition actually has changed a little bit. When I was an undergrad, we had a different definition, but this is the one that's sticking around for at least a decade now, so I'll stick with it. Yeah, the cytoplasm is everything inside the cell except the nucleus. So if this were a cell, identify the nucleus there. It's everything else that's inside here. What's that mean? It's going to mean cytosol, the juicy part. And your book's not going to call this fluid. It's going to call it a gel-like matrix or something like that. Think of it as fluid. Think of it as very watery. The image of a gel is not helpful for imagining how these processes happen. An image of water or watery fluid is actually more useful for imagining how processes happen inside the cell. And then organelles. That's our cytoplasm. Cytoskeleton. We've got a lot of information on the cytoskeleton. I really only want you to sort of know a couple of things. Let's spell this right. The cytoskeleton is basically like an infrastructure inside the cell. A good image to have is like building a skyscraper, and before they put like any real like walls and things on, there's